Validation of an expression vector for TTR gene. I would like to express protein transthyretin to make multiple copies of its DNA fragments for easier studies and manipulation. For example, in the study of ATTR amyloidosis, TTR gene encodes a protein, that's protein transthyretin, which carries tyrosine and retinol in the plasma and cerebrospinal fluid. This protein has also been found to be involved in cell functions like proteolysis, autophagy, nerve regeneration, glucose homeostasis. For this study, I will be using the PGMT vector, which utilizes CMV promoter to control the binding of RNA polymerase and transcription factors in the mammalian cell. Now, the restriction enzyme NTI that recognizes DNA sequence CATA-TG will be used to open the vector at the 592nd nucleotide. This same restriction enzyme will be used to open up the DNA of interest, which is the TTR. Now the DNA is then introduced into the vector by ligation. During this ligation, we use the T4 ligase 44. To transform bacteria, the plasmid containing the DNA itself is transferred into E. coli cell and to be sorted and replicated. Now, the E. coli cell will take up the plasmid that has been accumulated on the cell surface after it has been heated up at 40 degrees Celsius for 20 seconds. The transformed cells are plated on agar plates containing kanamycin, which is a selective antibiotic. Now, E. coli cells that have received the plasmid will then be able to grow into colonies on the agar plate because the plasmid that contains the gene insert has an antibiotic resistance gene. So it will be able to grow in the presence of kanamycin. That means most of the colonies formed on the agar plate as shown in the picture are the correct vectors. Now to validate if I have the correct vector, I will be using the Sanger sequencing method. Now the Sanger method has to do with unwinding the DNA, forming a single strand, and then adding a DNA primer, which marks where the sequencing will start by binding to the DNA strand. Now we had the three nucleotides and we had the enzyme DNA polymerase. This enzyme DNA polymerase will build a complementary DNA strand at the primary sequence by adding nucleotides one after the other. Now these special nucleotides are labeled a different color dye, so it is easy to read when exposed to UV light. The DNA polymerase will stop the DNA sequence it is building when it reaches a chain terminating nucleotide. That's why I have different lengths of DNA strands. So this DNA segment is placed under gel electrophoresis, whereby they will be separated by their sizes. Now the smaller size will go further than the larger ones, and then after it gets to a point where the pieces are no longer moving, then the gel is then placed under UV light below. Now we have an easier method to read the gel electrophoresis. We have the fluorophores, whereby we use a different color of fluorophore to combine to each nucleotide, and then they can be read at once. After the gene of interest has ligated with the vector with the DNA ligase to create a circle, it is then transfected into a mammalian cell, and we will be using HEK293 using electroporation. Now for this technique to be successful, the duration and strength of the electroporation must be optimized against the cell type. Now the cells and the DNA construct are suspended in the electroporation buffer, and then high pulse of electricity is then applied to the mixture. Now this pulse causes a potential difference across the cell membrane, and temporary pulse are introduced, which allows the exogenous DNA to enter into the cell. Now the transfected cells are then cultured and harvested by diluting the cells in a non-selective complete medium. Now for stable transformation, which is what I will be following, the cells are grown for 48 hours and transferred into an antibiotic containing medium. Thank you.